Okay, good morning, everybody. It's actually nice to be in person it's been a while. So it's my pleasure to introduce Leena Hilakivi-Clarke. She's a professor of food sciences and nutrition at the University of Minnesota and leader of women's health, the women's health section at the Hormel Institute. Leena has her PhD in experimental psychology and physiology from the University of Helsinki in Finland. After completing her PhD study, she was a Fogarty postdoctoral fellow at the National Institute of Alcohol Abuse and Alcoholism in Bethesda, Maryland. After that, she joined the Lombardi Comprehensive Cancer Center at Georgetown University in Washington, D.C., where she was until she joined the Hormel Institute and the University of Minnesota in August of 2020. She has a strong publication record consisting of over 160 journal articles. She's a recipient of multiple grant awards through her research career, including being a program director for NCI-funded U54 program project grant entitled Timing of Dietary Exposures and Breast Cancer Risk. And this was to investigate nutritional modulation of genetic pathways leading to breast cancer. Her research interests are many. They include possible connections between cancer and stress, as well as how maternal dietary exposures or obesity affect breast cancer risk in future generations. She is also interested in how obesity during pre- and postmenopause affect breast cancer risk, how diet influences breast cancer recurrence, and understanding mechanisms of how social isolation increases breast cancer risk and mortality. And she's also interested in prevention of cardiovascular diseases and postmenopausal breast cancer survivors on aromatase inhibitors. She is particularly interested in the role of gut microbiota in mediating the effects of diet on inflammation, tumor, immune microenvironment, response to immune checkpoint blockers, and breast cancer. And so the title of her talk today is Obesity, Social Isolation, and Breast Cancer, the Role of the Gut Microbiota. Please welcome Leena.
Thank you.
Thanks for joining us.
Thank you. And as you gathered, I have multiple interests, and so I had a little bit difficulties figuring out what I would want to talk to you today. And I chose to talk about obesity, social isolation, and breast cancer, and a little bit about gut microbiota. I know that you have several experts here who could be able to talk about the gut microbiota much more than I do. So I left it there at the very end. And I'm in a process of putting together your one grant about obesity and metabolic disturbances. And this talk is quite a lot about that. And so I would really appreciate your feedback if you feel that I say something funny or you have some clever ideas to suggest to me. And so you can leave it at the end, or you can just raise your hand. I might have difficulties for the people who are Zooming to see that you are asking. So I think that I need to leave that at the end. So obesity, social isolation, and breast cancer. We know that obesity increases the risk of many types of cancers, most endometrial cancer, but breast cancer is there too. Breast cancer and obesity association is very complex. And that is because we know that if a woman is born having a high birth weight, later on she's at increased risk of getting breast cancer. But if she's overweight or obese during childhood and premenopausal years, she's actually at reduced risk. I think that's mine. Should I turn it off or? It will stop, yes. Yeah. It normally doesn't ring, so that's why I forgot to turn it off. So premenopausal obesity protects against breast cancer, and it's only when a woman hits menopause then obesity starts increasing the risk. And you can see that well in these two figures. The first one is premenopausal and the second one is postmenopausal, and it's just comparing overweight and obese women. So the line in brown is obese women and the line in green is overweight. So you can see that premenopausal years, the heavier a woman is, the lower the risk. And the in the axis, x-axis, it's a waist-hip ratio, and that's a measure of central obesity. So if a woman is centrally obese during premenopausal years, but doesn't have necessarily very high body mass index, she's at an increased risk of getting breast cancer. And that actually reverses the protective effects of obesity. So it seems that the central obesity is an exception in terms of increasing premenopausal breast cancer. And here you can see that the fat storage changes at menopause. So premenopausal women have most of their fat stored as subcutaneous fat. 80% of the fat is there. But then 5 years from switching from premenopause to postmenopause, now 65% of the fat is subcutaneous. And when we gain weight, when we get older, it's more of the weight goes into visceral fat or central fat and less as a subcutaneous fat. And there is dramatic differences between the subcutaneous and visceral fat. And we know from the fact that the switch occurs at menopause that estrogens have something to do with protecting the body from getting the visceral fat. But there are other factors too. And here it's PGC1 alpha, and then another which I can't read very well because it's so small here, but I'm not going to talk about that. So just to show you that the visceral fat is located inside the body around the internal organs, and the subcutaneous fat is under the skin, and that's the fat that is in our butt, in the legs, in the arms, and so on. But this visceral and the central fat is around the waist. And you probably have heard about that some obese individuals have healthy fat, and that is because if they mostly have the subcutaneous fat, that helps to maintain the insulin sensitivity, and so its subcutaneous fat is okay. When it's in excess, then it starts to resemble a little bit more the visceral fat. But the visceral fat is the one that releases different cytokines, so it's highly inflammatory, and it also has lots of metabolic changes, so it can cause metabolic disturbances around the body by interacting with the other tissues. So why are some premenopausal women have the central obesity even though they are not otherwise heavy? So I looked at the literature, so what can protect against the visceral obesity, or what makes a premenopausal woman having visceral central obesity? It's kind of a silly. So being physically inactive, that makes sense. Not drinking coffee, so we should all drink coffee to avoid central obesity. Being married. Many of these studies were done in African countries where if you are married, then you are able to eat more and perhaps store the fat as a central fat. Being moderately or highly stressed is one of the factors that increases the visceral or central obesity. And here is a picture that you can't see that shows that how the stress and perhaps the cortisol leads to increased obesity. And then one factor you probably have heard is sleep. So people who sleep less than 5 hours a night are much more likely to develop central obesity than people who sleep 8 hours or more. So it is good advice to try to avoid stress, but unfortunately, stress is not linked to breast cancer or other cancers too. So it is a topic that has been studied a lot, and some people feel very passionately that there must be a link because there are these anecdotal stories how a person lost somebody or experienced strong work-related stress, and then the next thing we know that person is diagnosed with cancer. But in studies done, there doesn't seem to be a link. And here is an example of the Women's Health Initiative that in addition to looking stressful life events, also looked if a person had low social support or high social support. So the only group that had an increased breast cancer risk were the ones who had some stress in their life and didn't have any social support. But otherwise, I don't know how. Okay, otherwise you can see that the more stress, the lower the risk. So the only stress that increases breast cancer risk and mortality is social isolation stress. And we know that social isolation causes weight gain, and it also seems to be increasing the visceral fat deposits. So what is social isolation? When I started studying social isolation many years ago, and this is kind of a side project for me, people thought that social isolation means inmates who are kept in social isolation. No, social isolation can be defined in different ways. And because of the COVID-19, we all know what is one form of social isolation, but it is distancing of an individual psychologically or physically or both from his or her network of desired or needed relationships with other persons. And the incidence of loneliness is quite high in different countries. The ones in blue are the ones where the loneliness is highest. And I was happy to see that Finland was white, because I always thought that Finnish people are socially isolated. They kind of are, but that's the way of life, and perhaps the same for Minnesota. But no, I think here people are much more interacting with each other. So people keep to themselves, but it doesn't bother them, so they don't feel lonely. But people in the Middle Eastern or Middle European countries, Italy particularly, they want to interact with each other. So if they can't, they feel very lonely. So this became to the level of a problem, the social isolation or loneliness, that in the UK in 2018, when Theresa May was the prime minister, she decided that they needed to have a new minister for loneliness. And now there's a third person holding that position. She's the Baroness Diana Barron. And that was because there are so many older people in UK, and it's known that loneliness is related to lots of health problems. Interestingly, also the Surgeon General, current Surgeon General Vivek Murthy, when he was traveling around the country and talking about health, he got the impression that there were lots of lonely people, and that was related to their health. So he actually published a book, and that was published just when the COVID started running around, but it was written before the COVID. So who suffers from social isolation in addition to all of us because of the COVID? Older people are much more likely to do so. People who suffer from poverty, abuse, race, ethnicity, religion can cause social isolation, disability, depression, anxiety, obesity, and then any disease that, life-threatening disease that we are diagnosed with causes the feeling of being lonely, even though we are surrounded by people who, well-meaning people who want to help us, but you feel alone with the disease and alone with your future. The loneliness and social isolation has been assessed amongst breast cancer patients, and this particular assessment was done between 6 months and 2 years from diagnosis, and it represents 4 different studies, and those studies, these 16 to 41.2% of the patients could be characterized as socially isolated. So how bad is social isolation? It is equally bad as smoking or obesity in shortening the life. And here you can see social isolation, obesity, and smoking affecting all-cause mortality or breast cancer mortality. So it is clearly something that we should take seriously, but it is much more easy to talk about smoking and its adverse effects than something that is in our head, social isolation. How would it be able to affect us? But it does. And I'm going to talk a little bit how it does it. So in animal studies, there have been many animal studies looking if social isolation, and the social isolation in animals is housing them by themselves. The female mice and rats do not like at all being by themselves. They are very social animals. So if you house them by themselves, and that increases their risk of breast cancer. And this has been shown in many different models. Here, the study was done in wild rats, and the wild rats didn't get any carcinogens or anything. They just caught much more tumors about 2 years of age when they are very old than the ones that were grouped together or housed together. In humans, it's difficult to assess if social isolation increases the risk of cancer, because once you know, there are not necessary studies that have followed socially isolated people and see who gets cancer and who doesn't. It has been done using, for example, post-traumatic stress disorder as a symptom of social isolation, because it's one of the aspects of PTSD is social isolation. And a study done amongst nurses who served during Vietnam War and developed PTSD, they were 3 times more likely to develop breast cancer than nurses who didn't get PTSD. This has also been studied, for example, in Denmark, just PTSD and cancer risk, and there they didn't find 20% increase, but that wasn't significant. But there are several studies that have looked social isolation and cancer mortality. This study was done, I think, in Harvard, and they found that socially isolated men and women are 3.6 times more likely to die from cancer than socially integrated individuals. Then there are several studies that have looked breast cancer specifically. Most recent study shows that women who are diagnosed with early stage breast cancer, if they are socially isolated, they are 43% more likely to show recurrence and 64% more likely of dying of breast cancer than socially integrated breast cancer patients. In preclinical studies, it has been shown that mice respond less well to chemotherapy if they are kept in social isolation. And social isolation doesn't just affect the cancer, it increases type 2 diabetes, Alzheimer's disease, and one very much looked disease in terms of social isolation is cardiovascular diseases and stroke. And this is a study that involved almost 500,000 people in UK, and they looked if social isolation increased the risk of acute myocardial infarction or stroke, and it did. And they also found that the patients, if they got those and they were socially isolated, they were more likely to die of heart attack or stroke than socially integrated. And we know that social isolation is linked to other factors that makes people socially isolated, like being overweight or obese, or being poor, or having other factors. So they actually controlled all the different factors, and that's what is here. Different factors were controlled, and when all the factors were controlled, social isolation alone, regardless of the factors linked to social isolation, increased the risk of cardiovascular diseases and stroke. So then why does social isolation increase the risk of all these diseases and increase mortality? So it has been suggested that it is because of the cortisol, so stress response, or it's because of the adrenergic response. The problem is that the data is quite inconsistent regarding the adrenergic system, and then the same involves with cortisol or corticosteroid in the animal experiments. So there is initially an increase, but then in the longer term social isolation, there is no more increase in cortisol levels. And we also know that cortisol is anti-inflammatory, and it inhibits ER-positive breast cancer. It increases triple negative, but it's protective against ER-positive breast cancer. But I'm not quite certain what is the role of the cortisol in mediating effect of social isolation. So I just tell you my concerns, but I'm not telling that cannot be the pathway. So what other possibilities are there? So this is a study done with fruit flies, and some were kept by themselves in the glass jar, I assume, where they're kept, and some were with lots of other fruit flies. And they found that if they were kept by themselves, they slept poorly, they had high levels of inflammatory cytokines, and they had misfolded proteins. So the misfolded proteins could, and then they have also central obesity, if one can study that in the fly, but they were heavier and than the flies that were together. And so what does that all link to? So it could be mitochondrial dysfunction. We know that unfolded proteins can cause mitochondrial dysfunction, but mitochondrial dysfunction also can be inducing the dysfolded proteins. Definitely there is a link to inflammation, and then central obesity, we know that is related to impaired metabolism. So we wondered if there is a difference between obesity and social isolation in terms of affecting insulin sensitivity and breast cancer risk. So we did an animal study using C57 black mice. These are the mice that are highly susceptible for high-fat diet. They gain weight and become obese. And we group housed them or kept them by themselves and then divided them to two groups. One got control diet and one got obesity-inducing diet. And then we gave them DMPA to induce mammary tumors. We assessed their insulin sensitivity and then just monitored their mammary tumorigenesis. And this is the two different diets. So we had lard as the source of the high fat, but they were well matched. And the animals that were kept socially isolated, so one who got the obesity-inducing diet, they ate a little less food because the food is so energy dense. The animals that were socially isolated but got control food, they eat much more. And so did the socially isolated who were also getting the obesity-inducing diet. And the caloric intake went also was highest in the socially isolated high-fat diet fed, and the weight gain was highest in those mice. Then we looked if the typical fat-related factors were altered, and we found leptin levels were highly elevated in the obese animals that were group housed, but not so much in the socially isolated. Adiponectin levels were all down, but significantly so in the obese group housed. And PPR gamma that regulates the adipose differentiation, that was significantly downregulated in the socially isolated and obesity-inducing diet-fed mice. Oops. When we did the glucose sensitivity and insulin tolerance test, we found that the insulin intolerance was highest in the socially isolated and group housed animals, and was slightly higher in the obese than just socially isolated. The blood glucose tolerance was most significantly elevated in the socially isolated that got the regular diet. I don't know why this takes time to, okay. So the study showed that this is the mammary tumorigenesis. Skipped for some reason the slide. So we found that the incidence of mammary tumors was highest in the animals that were socially isolated and fed obesity-inducing diet. It was similar in the obese or socially isolated and higher than in the control group housed animals. So this was with the DMPA model. And so we want to study this further, and it would be great to have a model which doesn't take as long as a DMPA model. So we wondered if allografting tumor cells to mice that are socially isolated or group house would make them to grow faster. And there are a couple of studies in the literature using the 41 cells in pulp C mice. And the pulp C mice are very resistant to obesity, and they have a good metabolism. And in them, we didn't see an increase in the tumor growth. Have we looked metastasis, we probably would have seen that. But in the EO771 that are allografted to C57 black mice, there we saw that they grew bigger, faster than if they were socially isolated than if they were group housed. So back to the other study where we used DMPA as a tumor model. We also looked if there was any changes in the tumor histopathology, and there wasn't. And we looked if the ER alpha and PGR expression was modified by the different exposures, and they were not. Then we looked the unfolded protein response, and there we found that socially isolated animals had the highest level of GPR 78, indicating UPR was activated, and IRE1 alpha expression was also elevated. So I don't know whether you can read them. So this is the social isolation and obesity-inducing diet, and this is the obesity-inducing diet alone. So we also wanted to look at the mammary tissue and adipose tissue level, what happens to one of the key genes related to or preventing subcutaneous fat from being converted to visceral fat. And so they were the group housed, obese, socially isolated and obese. And you can see, as one would expect, that obesity and social isolation with the fat increase the size of the fat cells in the visceral abdominal adipose tissue and somewhat in the mammary adipose tissue. And when we looked the PGC1 alpha levels, they were dramatically reduced in the socially isolated and obese, both in the adipose fat and mammary gland. So for us, this feels that it's an indication that social isolation promotes visceral adipose tissue. So then we did another study, because lots of the human studies have been looking breast cancer mortality and social isolation. So we wanted to see if also in the animal models, the social isolation would affect the response to antiestrogen treatment, or perhaps recurrence after the treatment is stopped. And so we did a study in rats, because they get ER alpha positive tumors, mammary tumors that respond to tamoxifen. So we, when the animals have a detectable tumor, we divided them to socially isolated or group housed, and then started feeding them tamoxifen when the tumor was a specific size, and looked if they showed complete response to tamoxifen, partial response, the animal never responded to tamoxifen or the tumor, or then they acquired resistance to tamoxifen and developed a new tumor whilst they were being treated. And animals that showed complete response to tamoxifen for 9 weeks, which is 5 years of human life, we took tamoxifen out and then monitored the animals if the tumor might come back. And we found, in terms of response to tamoxifen, that there was no difference. Social isolation didn't impair the response to tamoxifen. There was a little bit less complete responses, 31 in the group housed and only 21 in the socially isolated, but that wasn't significant. Then when we looked recurrences after tamoxifen, there was a increase from 33 reoccurring in the group housed to 60 reoccurring, so almost double increase in reoccurrences in the socially isolated after tamoxifen. And here you can see it nicely. This is during tamoxifen, the tumor burden in the animal, tamoxifen out, socially isolated animals start showing a high tumor burden. So now we wanted then to explore what are the changes that take place in the mammary gland that could be responsible for the differences in reoccurrence and perhaps differences in risk of getting breast cancer. And we did RNA sequencing, and we were discussing, should we use tumors or should we use normal mammary glands? And we decided that we will use normal mammary glands, because if we had used tumors, so for example, we had to have to use tumors that didn't respond to tamoxifen, so we would be comparing resistant tumor to another resistant tumor. So we thought it's best to do it from the mammary glands. And we found with using the GSEA, here are the top 50 up and top 50 down-regulated genes. And if you look at what is the top down-regulated gene, it's this BROS that when it's down-regulated increases cardiovascular deaths. And so these were in the mammary gland. And then the top upregulated genes were related to glutamate action, and they are all linked to schizophrenia. And we know that social isolation is the animal model of schizophrenia. So it was interesting to see these changes in the mammary gland. Okay, then when we did the co-pathway analysis, looked what pathways were altered, it seems that the changes were indicative of increased cell proliferation. And we had actually looked cell proliferation in a previous study where we fed them obesity-inducing diet and kept them socially isolated. And there it seems that there was more proliferation in the animals that were socially isolated and get the obesity-inducing diet compared to if they only got the obesity-inducing diet. And there seemed to be a little less apoptosis, but that wasn't, it seems quite dramatic, but it's not significant. Then when we put all the genes to cancer hallmark pathways or KEX signaling pathways, we found that the changes that what was suppressed in socially isolated animals was oxidative phosphorylation, and what was upregulated was IL-6, JAK-STAT3 pathway, and lots of other inflammatory pathways, both in the cancer hallmark and KEX signaling analysis. Here's the oxidative phosphorylation, and here are tons of different inflammatory pathways. And here you can see better in the cancer hallmark pathway. This is IL-6, JAK-STAT3 pathway analysis. So it was significantly increased in socially isolated, and oxidative phosphorylation was significantly reduced in the socially isolated. And here are just some of the genes that were in those pathways. And we confirmed many of the oxidative phosphorylation signaling genes so that they were all downregulated in the socially isolated. We were interested in whether these were also downregulated in the obese animals that were group housed, and yes, they were. And so mice is kind of an interesting in comparison to humans. When you feed mice high-fat diet, they gain both visceral and subcutaneous fat, even though they have still their ovarian function. But their estrogen levels are probably too low to have them to have more subcutaneous than visceral fat when they are fed obesity-inducing diet. So, and when, So
Leena, if you could just repeat that question so our folks on Zoom.
So how long were the animals in social isolation? So in the study where we looked the obesity and social isolation, they were socially isolated from week 4 onwards. And we monitored their tumorigenesis for 20 weeks after they got their last carcinogen exposure at age 11 weeks. So they were a long time. In the antiestrogen study, we actually did a study in mice where we started at the same time and didn't see any change in the response to antiestrogens. We were thinking, oh, perhaps it's because we kept them too long. So in the rat study, we started the social isolation at the time they started tamoxifen, or like a couple of weeks before.
So the gene expression study was
in the rats.
Okay.
And they were collected after they have completed tamoxifen and were 9 weeks off from tamoxifen.
Thank you.
So it was a very, very long-term social isolation. So when we looked what pathways were involved in the oxidative phosphorylation suppression, it was basically everything. It was the enzyme in the electron in the oxidative phosphorylation pathway, but also glycolysis pathway. This MPC was downregulated, so suggesting less pyruvate got back. There were several genes in the TCA pathway that were all suppressed. And I go back to that. So oftentimes we know that oxidative phosphorylation goes down and the pyruvate goes to lactate instead, and that's the Warburg effect. So we were curious if the Warburg effect would have been activated in the mammary gland of the socially isolated animals, but it wasn't. And then when we looked the inflammatory pathways, we found, several. We were able to confirm several genes, and we also looked then in the mammary tumors that IL-6 was significantly up, and phosphorylated STAT3 was significantly up in the socially isolated animals. And we have recently also started looking what changes might happen in the tumor microenvironment, particularly in the immune cells. And Fabia, who is a postdoc, or now she's a senior scientist in my lab, has been doing flow assays. And what she found that the myeloid-derived suppressor cells that are the key immunosuppressive cells in the tumor microenvironment, they were significantly increased in the socially isolated animals. And this was in the spleen. We have just started doing them, so we have only assessed in spleen thus far. So it seems that there are these two pathways that are most affected by social isolation. It's the suppression of oxidative phosphorylation and increase in inflammatory pathways. Interestingly, these two pathways interact with each other. Inflammation can cause suppression of oxidative phosphorylation, but oxidative phosphorylation also can cause inflammation. So that would be interesting to figure out which one is the chicken and which one is the egg. We don't know yet. So we can also then wonder, how does the social isolation affect these inflammatory and oxidative phosphorylation pathways? Because it is social isolation. It is something that happens in our head initially. And so one obvious change would be the gut microbiota, because the brain-gut axis. And we know that like cortisol can affect the gut microbiota and inflammatory changes, and the enteric pathway can affect also lots of these things. I'm going to skip that. And if we think about the mitochondria, the metabolites of the gut bacteria affect. So there's tons of different bacteria that are involved in different metabolic pathways that all affect the oxidative phosphorylation and TCA cycle. And that actually the changes in the TCA cycle can also affect the epigenetic regulation. And I don't have time to talk about the epigenetic regulation, but that is something that social isolation also seems to be inducing HDAC and DNMT upregulation. So what is altered in the gut microbiota of obese individuals? So there are several bacteria that has been found that are up, or these are down, and perhaps mostly people talk about the firmicutes to bacteroides ratio that is increased in obesity. So how bad these are, it's not clear to me, because if one just focuses on single bacteria species or even this ratio, the data is all over the place, what is bad and what is good. For example, Prolifera, that is higher in lean individuals, and it's not necessarily always the best of the bacteria. And we know that if one treats people with immunocheckpoint blockers, the ones who have high level of bacteria that is increased by obesity respond best to the therapy, and the ones who have high levels of the ones, like, for example, the Prolifera, are the ones that respond most poorly to these immunocheckpoint blockers. And obesity is one of the factors that make individuals respond better to the immune checkpoint blockers. So there might be some validity. But also the interaction between obesity and gut microbiota is an interesting one, because so I assume that it's what we eat affects the gut microbiota, but it might also be that the gut microbiota affects obesity. And those studies, you're probably familiar, if gut microbiota was transferred to a mouse, lean mouse, from an obese individual, that mouse then became obese. But if, and these were identical twins that were used in the study. So another identical twin that wasn't obese, his microbiota was transferred to another mouse, and that mouse stayed lean. So clearly gut microbiota and obesity affect each other. In terms of social isolation and gut microbiota, there are basically no studies, I would say. There is one study which I'm going to show to you, but then there are lots of studies related to gut microbiota and autism. And we can think autism as an ultimate social isolation, because that's very typical for autism to not to be socially integrated or live in their own head more than be socially interactive. And so there was an animal study published in Cell a couple of years ago, where they used maternal obesity as a way to make the mice autistic, and they found that the gut microbiota was altered in the offspring, autistic offspring of obese mothers. And when they then gave these offspring the bacteria that they were missing, then the autistic mice became more socially interactive. And in autism now there is some evidence that treating them in addition to other therapies with specific probiotics will help with the symptoms. But that's basically it. Then there was one study that was published 2020 that involved prairie voles that are very social animals, and some of them were kept in isolation for 6 weeks, and their stools were collected before and after isolation, and they found several changes in them. They looked females and males, but based on one study, it's really difficult to figure out what's going on. We have done a pilot study where we just collected poop after the social isolation or group housing, and we found that Akkermansia municipala was very significantly reduced, and verrocomicropia in the socially isolated animals. And we also found that the Lachnospira different species were increased in socially isolated animals. So what would that mean? I don't know. And when we compared the firmicosides to bactericides, we found that social isolation increased the abundance of the firmicosides and reduced the abundance of bactericides. So in the same way, what happens in obesity. And then the Akkermansia municipal was significantly down, which also is down in obesity. So there we did another study where we are comparing the gut microbiota to inflammatory markers, and Chris Daly, his lab is doing the assay. I think they have done them, but we haven't yet got the data. So that's an interesting, perhaps more better study than our little pilot study, where we just wanted to see if socially isolated animals have different composition of the gut microbiota. So to summarize, central obesity in premenopausal women increases breast cancer risk. It is important to identify the factors causing central obesity and eliminate them. We found that social isolation inhibits PPR gamma and PGC1 alpha that are switches to block saturated subcutaneous adipose tissue to being stored as visceral adipose tissue. And thus it can be one of the causes of central obesity. The two key pathways that were affected by social isolation was an increase in IL-6, STAT3 signaling, and suppression of mitochondrial oxidative phosphorylation. And social isolation might increase inflammation and impair mitochondrial oxidative phosphorylation by altering the gut microbiota. So future studies are needed to figure this out. And so if you are wondering how could one actually reduce the visceral obesity in premenopausal women and postmenopausal women too, there are some, like, physical activity is very effective in reducing visceral fat. It's not very effective for subcutaneous fat. And then there are some, like, plant-derived compounds that have been shown to be effective, that are very high levels of raspberries and blackberries. And then ketogenic diet seems to be also very good in affecting both. So there was, most of the studies were done at Georgetown, and so I have many people to thank for these studies. I don't have too many people or organizations who have funded this, because it still remains like social isolation. Who cares? Even though it is as important as smoking and obesity in killing us. Thank you.
So that was a lovely talk. We can open it to questions.
I have a quick question about the tumor models. I'm not that familiar with the tumors that grow up in these DMBA. Are they like the rat? Do they have ER positive tumors?
Yes.
They do. Okay.
And they are nice because they are the only model that I know that respond to antiestrogens.
Yeah.
You can't really test the aromatase inhibitors because the animals don't have the mammary aromatase.
Interesting. Okay.
enzyme.
Great. Anyone else from the room?
I don't currently see any questions coming through the chat function right now from our group. Are there any questions from our Zoom audience?
While we're waiting for the Zoom audience, I was wondering how you envision translating some of your findings to humans. I was wondering how you were thinking about translating your findings to humans. Because
So I would be very interested in looking premenopausal women in terms of the social isolation and the visceral versus subcutaneous fat. Subcutaneous fat, we can easily collect. Visceral fat is more complex, but so perhaps figuring out if the results are also true for humans, and perhaps do some human tissue cell culture combination, and then looking particularly if we know that social isolation increases the inflammatory cytokines in the circulation, but nobody has necessarily looked the oxidative phosphorylation and the mitochondrial function. Yes, and then just looking if eating the raspberries and blackberries or being in ketogenic diet would be helpful.
All right, great. We have one question from our Zoom group from Doug Yee. How do you account for different personalities in humans? Some people are okay with social isolation, while others are more gregarious and need human contact.
So there are several different tools that are used to assess social isolation and loneliness. So, and those take into account the fact that some people like being solitude, and some people don't like it. It's the level of distress that is caused by not having the contacts. And so even if one is okay with being alone, it has been shown that people who, like older people, they are much more likely to get different diseases if they are by themselves than if they have a partner or friends. So sometimes it's what one feels, and sometimes it's just the presence of the interaction, even if they sometimes don't feel very good to, why are you yapping?
So does the use of social media and computers kind of throw a wrench into the model at all? I mean, do you have to factor in how much time people spend on Facebook because they're physically socially isolated, but not emotionally, maybe if they're online with all their friends?
So that's not my area of expertise, but I'm pretty sure they are getting into that also when they are assessing. But it's known that the social isolation has increased in young people because of they play games and stare at the computers.
Yeah, yep, and don't move around.
Yes, yeah.
It's fascinating. Thank you. I'll give this back to Lisa.
Thank you.